he went in one day and he looked and he saw that there was, in this one tank, all the sea lamprey were dead at the bottom, and all the other fish were just zipping around perfectly fine. I was
That's
like, oh my
awesome.
god, this is
Interesting.
crazy. And that was a chemical called TFM.
Hello, everyone, and welcome into the Chasing Wild podcast, your home for all the best content on conservation, wildlife, and adventure. Our mission is to bring the wild to you. We are your hosts, Jake Johnson, Dean Dudzik, and today we have a special guest, one of my best friends, Connor O'Kane. Connor, how you doing?
Doing all right. How are you guys?
Doing good, doing good. Happy to have you
Yeah.
on the show. So Connor O'Kane, Connor is again a friend of mine. We went to school together at Miami University. We roomed together for 3 years, got his degree in biology, and then he was most recently at the Michigan State, right? Michigan State master's
Mhm.
program, getting his master's degree in fisheries. Today we're going to be talking a little bit about his program with Michigan State, where he focused on sea lamprey. And before we jump into that, Connor, why don't you just go ahead and introduce yourself, give us a little bit about you, and maybe some cardistry talk a little bit. So,
Ooh, a little cardistry talk. Yeah, I can do that. So I grew up around the Chicago area, so similar to Jake as well. Went to, as you mentioned, Miami University, got a degree in biology. After then, I spent some time focusing more on marine and aquatic sciences. So I spent some time at a couple research technician places around Chicago, looking at aquatic technician, working on aquatic technician, fisheries technician type work, as well as the shed aquarium as well. Spent a little bit of time there. And then I eventually got into sort of the genetic side of things. So I sort of spent some time self-teaching a bit and getting more interested in genetics, and that led me into wanting to jump into a master's program and really kind of do some of my own research. So that led me to Michigan State, and it led me to the thesis project that I worked on, which I'm sure we'll get into a little bit. And then outside of schooling and jobs and interests, I do a little bit of this thing called cardistry, which normally I have to explain it to people without showing them, but we do a video camera here, so I suppose it's just a little easier to maybe grab a deck of cards. Usually have one lying around, not too far
they're never
from
too
me,
far from
never
you.
too
Yeah,
far.
never too far.
Um, but you can think of it with like a magic trick. Sometimes, well, let's see, I could bring my camera down a little. With a magic trick, sometimes they might have a fancy shuffle as a part of the trick or something like that, but nothing too much. But what cardistry is it takes those shuffles and it turns it into the trick itself. So the fancy shuffling of a deck is the trick. So there's nothing, I'm not trying to trick you. I'm just showing you just a skill-based thing. Hopefully looks kind of cool if you practice it enough. And I've been doing it for about 12 or 13 years at this point. And I actually started my own brand with it. So this deck right here, quick plug, Square Up cards.
Oh
Uh,
yeah, man.
it's my
No,
own brand that I started, and with a couple of my good friends as well. But yeah, that's cardistry. Just sort of fancy shuffling cards, and one of the things I do in my free time.
Dean, you got anything for him there? Any questions about the cardistry? I know it's not as familiar for a lot of people.
hmm So it's not a magic trick. That's what you're telling me.
Yeah, it started with false shuffles. So you might have, say, Jack of Spades is on top, and then I'll tell you that I'll go ahead and I'll shuffle your Jack of Spades into the center of the deck here.
hmm
But really, I just kept it on top the whole time. So that's kind of where
Ah,
it started to blend in from
yeah,
Magic, where that would be part of Magic Trick, and then people just started to do the shuffling all on its own, and I might
that's awesome.
teach that
Okay.
online. And there's a big thing is creating your moves and teaching them online to other
Yeah.
people. And the better moves you create, the more kind of credit you get.
Yeah. So it's like,
Street cred in a way.
yeah, it's very technique based.
Technique based and how well you can perform and how smooth you can be.
Yeah.
And then usually you'll create maybe 10 moves or so, practice them really well, and you'll put them all into a performance video. So you'll have a 3 or so minute long video. You can, you're in charge of all the art direction. You can make it crazy. You can make it really simple. And then you'll put that online. So sort of like how a musical artist might have a collection of 10 songs. So there's put it into an album and release that all together. And
Sure.
then other people can watch it and be like, oh, this guy's really good, or that move is a crazy idea. This guy's really good, stuff like that.
Yeah.
But yeah,
And
it's a
I
really
think,
cool community.
yeah, and I think to all non-cardists, every move looks cool because we're just like, what? I remember when we started living together sophomore year, you always were messing with a card deck while you're studying or doing it, literally anything. And I was like, what is this guy doing, man? This is crazy. But yeah, Square Up is Connor's company. Make sure you check it out. Lots of cool content there, especially if you're into like Instagram reels and stuff. A lot of cool videos. And Connor does a lot of the editing and videography himself, as well as with his friend Dom. Dom's last name is
Corrado.
Corrado. Dom Corrado. Great company, a lot of cool stuff, and they work very hard at what they do. So love it. Way to plug in the beginning. So let's jump into your master's program. So a sea lamprey, as simple as it can be. What is it? Why are you focusing on this?
Yeah, so sea lampreys are these interesting little creatures. They're a type of fish. They don't quite look like your typical fish. They look like an eel in a way, but instead of the typical mouth on an eel that opens and closes, they're actually a interesting group of animals called jawless fishes. So they don't have a full jaw. They have this circular disc, and it's full of rows of teeth. So they look like something out of a horror movie.
Hmm.
They can get 1, 2 feet long, maybe a little longer. And what they do is they attach to the side of another fish and they feed on the blood of that fish. So thinking of them like a leech in a way. So they're parasitic and they can sometimes kill their hosts, sometimes they don't kill their hosts. That's sort of uh, the interest in sea lampreys comes from them having initially evolved in the Atlantic Ocean, and their native range stretches all the way to the Mediterranean. So they evolved with these larger fish that they would feed on, and if you're a good parasite, you won't kill your host. You want your host to survive so that you can also survive. Um, However, that changed when there was the creation of the Welland Canal, and that bypasses Niagara Falls. So sea lamprey were then able to migrate into the Great Lakes. This was in the early 1900s, and they spread pretty quickly throughout the Great Lakes. And but that difference in the type of fish that they evolved to feed on was noticeable in the Great Lakes because the Great Lakes, the fish were smaller. So the native predators, such as lake trout, there were smaller than those oceanic fish they'd feed on, and oftentimes lamprey would then kill their hosts. So that sort of led to this huge issue of sea lamprey being this invasive species. It was putting a lot of pressure on the native populations of fish in the Great Lakes, uh, and it led to all these conservation and management efforts, uh, eventually stemming into some of the work that I did in my thesis. But yeah.
Yeah, and
Very
I
cool.
think it's really interesting because, when you think about, honestly, I was doing some research on the sea lamprey ahead of time, just kind of, I obviously know about your program, we talked about it a few times, but just, going back and looking up on them, when you look up sea lamprey, there's like hardly, I mean, you can find it, but there's not as much information about them in their native range as there is like every article or like anything I'm seeing is all about how significant they are as an invasive species in the Great Lakes because it's become such a problem. If Maddie, my wife, so I was talking to her beforehand, she's always shown great interest in your project. She was wondering, and I'm wondering the same thing, is there an end to sea lamprey in the Great Lakes? is there some sort of way? How is the conservation of removing them and or reducing them evolved, and how can we essentially manage this population in the Great Lakes?
Yeah, fantastic question. So yeah, that sort of picks up where I left off. There was you have this
Hmm.
invasion of sea lamprey into the Great Lakes, right? So the predation of sea lamprey on the native fish, particularly lake trout and some other fish such as whitefish, was detrimental to these populations, combined with a lot of the commercial and recreational fishing that was going on of lake trout and other species. Uh, Lake trout populations declined significantly. So there was a whole effort on, okay, we need to do something about this. The commercial fishery for lake trout in the Great Lakes more or less collapsed. So that's why there was such management efforts and control efforts involved in this.
Hmm.
And there were some initial efforts, such as physical barriers, physical traps, to prevent sea lamprey from migrating and spawning in the Great Lakes. So, as a quick aside on sea lamprey reproductive cycle, you can think of sea lamprey as similar to salmon in the way that they'll spend their life, or the main phase of their life, in the Great Lakes, in our case, in the Atlantic Ocean, in their native range. But in our case, we're talking about the Great Lakes,
Hmm.
so they'll spend this parasitic phase in the Great Lakes. And then when they've sort of had their fill of the fish, they'll migrate upstream to spawn. So different tributaries throughout the region, and then they'll spawn. It's a singular event, so they'll spawn and die, and then after that, the larval lamprey spend about 3 to 7 years in the substrate of the streams that they're in. So they actually spend
Wow.
the majority of their life living in the substrate in the sand that is in these rivers and tributaries along the basin. After that, they'll metamorphose. So they spend in the sand, they're filter feeding. So they don't quite have the full jawless sort of circular disc that they use to feed on other fish. They'll metamorphose, they'll migrate downstream into the Great Lakes. From there, they'll spend about a year to a year and a half feeding on the fish in the Great Lakes, and then the cycle sort of repeats. So there were efforts to trap and block them from these spawning events. If you can stop them from either migrating upstream to spawn or migrating downstream into the lake, ideally that can sort of reduce a bit of the population. But a larger effort was undertaken to develop a chemical that could really reduce the sea lamprey population. And I was sort of spurred on by this guy, Vernon Applegate. He was a guy out of the University of Michigan in the 50s. And he went up to the northern tip of Michigan at this research center called Hammond Bay Biological Station, which is where currently one of my advisors works as well.
Hmm.
And he and his team set out to find some chemical that would only kill or harm sea lamprey and no other fish. So you can imagine they have this setup in these labs of rows of tiny little tanks where they have sea lamprey in some, regular fish in others, and then both fish, probably both fish in most, where they have sea lamprey along with other, you know, juvenile lake trout or bluegill or other fish. And they tested thousands of chemicals. They're just putting chemicals in this, see which fish die. Essentially, put another chemical and another chemical, and monitoring it. And it was around the 5,000th, 200th or so chemical that they tested. And they went in, and he went in one day and he looked and he saw that there was, in this one tank, all the sea lamprey were dead at the bottom, and all the other fish were just zipping around perfectly fine. I was
That's
like, oh my
awesome.
god,
It's
this is
interesting.
crazy. And that was a chemical called TFM, and I will spare myself from trying to remember the actual full chemical name here, but TFM is the abbreviation for it there, and that's what we use to this day. There's specific streams and tributaries that we have identified as sort of hotbeds for sea
Mm.
lamprey spawning, and we'll release that chemical into the streams there. And we've been doing this for a long time, and that's probably the primary way that we've controlled sea lamprey so far in the Great Lakes, and I, I want to say there's since the peak abundance in the mid 1900s, there's been about a 90% reduction in the sea lamprey population so far. So it's much more manageable at this point. We don't have a fully collapsed fishery in the sense. There's still a lot of recovery efforts and there's still a lot of management and control. It's still a huge process and it costs a lot of money every year to do so, but we've certainly made a lot of progress in that sense.
Yeah,
Interesting.
definitely interesting. Dean, you
So
got something?
yeah, so these invasive populations of sea lamprey, just to, if I'm understanding correctly, um, do the adult populations um, just live in the Great Lakes, or do they return to the ocean when they reach like sexual maturity? And like, how are the invasive populations as far as where they live different from the native populations?
Yeah, good question. The sea lamprey that live and reside in the Great Lakes, they'll just migrate up Great Lakes tributaries and then they'll go back
Okay.
down into the Great Lakes. It would be a long journey, sort of, to go back. But honestly, as far as how the sort of life stages and general biology and ecology of sea lamprey in their native range versus non-native range, I'm not too familiar on that. That is interesting to think about if there's any differences there, and I would imagine that would be an interesting point of study to see how may have sea lamprey adapted in one way or another to the Great Lakes that possibly made them a better predator or possibly contributed to the impacts that they've had on the Great Lakes. So that'll be interesting
Okay.
to look into, though.
Yeah, and it's that makes sense. That there's that lack of information that we kind of touched on earlier on like the
Mm-hmm.
native range and population of the sea lamprey.
Mm-hmm.
I would draw like a parallel to maybe we hear on the news a lot, like here in the US about invasive Burmese pythons and the level of research done on that species and their invasive range versus their native range is probably extremely lopsided. So it makes sense why there's like this wealth of information on sea lamprey in the Great Lakes, which is kind of ironic because that's not where they should naturally inhabit. So yeah, I was just wondering because I think that information you shared on their life cycle and everything was really interesting, and that was all new to me. Um, my only reference point for sea lamprey or seeing them at Chet Aquarium, they
Mm-hmm.
have
Mm-hmm.
I remember they have a little tank, a little exhibit in the corner in one of the aquarium sections, and I would say like the closest thing I could compare them to is if you guys have seen Pirates of the Caribbean, the Kraken in Pirates of the Caribbean,
Mm-hmm. Yeah.
and it
Yeah.
opens. It's I won't say it's Jaws because that's not the proper terminology, but it opens that mouth opening and
Mm.
it looks that's what the sea lamprey look like. It's like a bunch of rows of teeth and just like Connor described this huge disc that's obviously not a hinged jaw, but yeah, that's what they look like as far as I'm concerned. So they're really interesting.
Yeah,
Yeah, and I
they're.
always think with invasive species, it's always easy to paint like a good versus bad. Even lionfish, where they're invasive, they're extremely,
Hmm.
harmful. But it's just about where they're supposed to be, And it's interesting how a Burmese python, a sea lamprey, a zebra mussel can just wreak havoc in an environment that it's not supposed to be in. And yeah, I did see something to like 90%. Um, Decreasing their population by 90% is huge. That's a really big effort,
That's
especially
massive.
with that chemical coming into play. I didn't know that.
Yeah.
I think we had talked about the barriers that were put in place, and it seems like a really effective tool because they do migrate up those tributaries. So like being able to block that, but nothing's perfect. So double whammy, hit them with the barrier, hit them with the chemical, just zap them right there. But no, it's really interesting, and I thought it was super interesting that sea lamprey were around before the dinosaurs. Like they have been around forever, like 400 million years or something like that. Like crazy. They've been around on this planet forever, which is why their bodies are also made of cartilage, just like a shark. And I was reading they do feed on a lot of sharks and rays. So, very fascinating species. Again, just kind of going into more of like the not a good or bad. Each species kind of has its own thing, but it's just where it's meant to be. So, um, Connor, you did a lot, and you currently are looking to do more on genetics and more data analysis like that. So what did your project look on a day-to-day basis doing kind of this genetic analysis of the sea lamprey? Like, what are you looking at for?
Yeah, exactly. So that's all sort of the ecology background side of things, and then there's
Hmm.
so the other half of my work, is the molecular side of things. I was in the molecular ecology lab, so we sort of blend these two fields a bit. And there's still a lot we don't know about sea lamprey, even some things that you'd think would be sort of obvious or things that we would know about other species. Like you were saying, even the differences between the native and non-native ranges. Uh, we still don't know a lot about even the invasive species, things that they do in their day-to-day lives, essentially. Some basic questions such as how long they might attach to fish, where they kind of spend their time in the basin, because we really don't get a lot of direct observations on these fish. And one of the things that is we don't have a comprehensive understanding yet is what they're fully feeding on. Uh, if you have a, a non-blood feeding fish, you can think of a regular fish like a lake trout or something. If you want to know what it feeds on, there's a couple ways to do that. You can either catch a lot of lake trout and you can dissect them, open their stomach, and look at the stomach contents there, and you'll have either full fish that you can look at and say, oh, that's this type of fish, or you'll see some bones or other hard structures. Even in non-fish, if you're looking, you can identify the types of plants, or maybe there's some shells that the animal is eating. So these specific structures that survive in the stomach for some time, or you can sometimes use sort of like a water pressure to sort of scoop the contents of the stomach out, so without killing the animal and the fish as well, is another way to just look at those stomach contents. But it's a bit different with sea lamprey because they feed solely on blood. So if you catch a sea lamprey and dissect its stomach, you just have a bunch of blood sitting there, and there's no real identifiable structures to say, oh, this lamprey fed on this fish or that fish, etc. So the current way that we estimate what sea lamprey are feeding on is either a direct observation, so this would be via bycatch. So say we have some commercial fishermen going out into the lake, they're trying to catch some lake trout, and they set out a net to trawl, and they go along the lake and they just scoop up whatever goes in this net. Ideally, they're catching a lot of lake trout because that's what they're going for, but there's going to be other things that show up in that net. So anything that shows up in that net they're not looking for is called bycatch. And sometimes you can catch sea lamprey in that net, particularly if they're still attached to a lake trout. So bycatch and seeing what lamprey are currently attached to in your net is one way we know what they feed on. Another way is that even if they fed on something and detached before you caught that fish, they'll still leave this circular mark on the fish that it was feeding on. So say you're going out and catching whitefish, for example, and you catch a bunch of whitefish, the fisherman can monitor and take a look at, oh, this whitefish had a sea lamprey mark on it, so the sea lamprey must have fed on this fish here. And we have sort of these direct and indirect observations that we use to estimate and sort of get a picture of what sea lamprey are feeding on. But there's some issues that come along with that prevent us from getting the full picture. Some of that being, for example, if let's say there's a species that does not survive sea lamprey attacks very often. They very commonly are killed by the sea lamprey, and then they die and then they sink. Those fish, those species are not going to really be captured in the nets. So any fish that die from a sea lamprey attack don't get caught. So it doesn't get recorded that sea lamprey fed on this fish. And then of course any other direct observation of in the stomach with blood is not possible as well. So it can be sort of difficult compared to other fish to get this picture of what they're feeding on. So we took a genetic approach in my lab to essentially map the DNA that's in the sea lamprey stomach to the two different fish, and then presumably if the DNA of some fish is in the stomach of the sea lamprey would have been feeding on that fish. So that's a technique called DNA metabarcoding. And what it does is essentially you could think of, let's say we take a gene region. So we have the full genome, which is all of the genes, all the base pairs per organism. There are certain areas of that genome, we can call them gene markers, that are really good at identifying and being specific to a species. So they essentially mutate really quickly. So they quickly diverge from other species and become different. And so they can act in as a barcode in a way where we can amplify the DNA in the stomach of the sea lamprey, and we look at this specific gene region and amplify all the genes within that region. And ideally, they'll all be a little bit different from each other, and we can take those, map that to a database of all of the known sequences. So say we have a database of lake trout, whitefish, salmon, etc., and we say this gene region looks like this for lake trout, it looks like this for salmon, it looks like this for whitefish. We can then take our sample, see what we have in that sample, and say, oh, we have a couple hits for lake trout. Oh, there's a hit here for salmon. Oh, there's a lot for whitefish. This sea lamprey maybe was feeding on a lot of whitefish here. And that's essentially what the DNA metabarcoding is, and then applying it specifically to sea lamprey was my project. It's been applied to other organisms before. It's been applied to plenty of non-blood feeding species, and it's been applied to blood feeding species as well. So leeches, for example, it's been applied to mosquitoes, ticks are other examples of organisms that have used this. Other researchers have used this technique to figure out what those animals, what those insects are feeding on, but it hadn't quite been applied to sea lamprey yet. So my project was using that technique, trying to figure out if we can. We took samples of sea lamprey across the Great Lakes, and then the goal was to sort of get a picture of what sea lamprey are feeding on, sort of A, to see does this technique work with sea lamprey, and then B, to sort of get a little bit of an insight into what they're feeding on so far.
Woof, woof.
Yeah,
That's a quick
need any explanation?
genetics lesson.
No, yeah, I mean, if anyone out there took molecular biology and was like, what's going on? Yeah, I mean, this is why we need so many different levels of scientists and people just like in the field biology. This is a field biology thing, working to understand understand the animals, right? You're trying to understand the sea lamprey, what they're feeding on, and then also how to address the issues or the concerns that are going on. So super cool. When you guys find a sea lamprey like stuck in some like amber or something, like some like a mosquito, can you do me a favor and let me know what historic blood is in there? Because that's pretty cool. No, I'm just messing around. But you got anything for that one, Dean?
Well, I just wanted to say, Connor, you broke your genetics lesson down very well, in a way that many professors can't break down as simply. So, props to you for that.
Appreciate it.
That was a nice simplified genetics lesson. So when you were wrapping up, you obviously the goal of the research, like you said, was to find out what the sea lamprey among the Great Lakes are feeding on. So, were you able to identify the prey or host species based on the genetic samples? And B, was that method successful in your project?
Yeah. Overall, it was a successful method. There was a bit of a bit of a roadblock up first. The project was sort of laid out for me when I started my master's degree, uh, and we started the project and we were working on everything, ready to extract the DNA, amplify it, see what's in there, and sort of what we had kind of we touched on it earlier. Sea lamprey are very unique. They're a very unique species. They're this ancient prehistoric species. There's nothing the I won't say nothing, but there's very little related to them taxonomically currently.
Right.
I think the closest is a hagfish, but
Mhm.
those aren't in the Great Lakes. So really, when you think of fish in the Great Lakes, there's really nothing very close to it taxonomically, and that helps us out as far as we want to be able to suss out what's sea lamprey, right? Because we don't need sea lamprey DNA. We know there's going to be sea lamprey DNA in the stomach, uh,
Right.
and it helps us separate that from the other fish. We thought it wouldn't be an issue first, but it turns out when we were amplifying all the DNA in the stomach, we were getting the vast majority of uh, the DNA that comes back and is amplified. It's called sequence read counts. The vast majority of those counts were sea lamprey, and a tiny bit was what we were interested in, was the fish
Mhm.
in the sample. So, sort of an addition to my project was we developed this called a blocking primer, which essentially blocks the amplifying of sea lamprey DNA, but it allows the amplification of all the other DNA in the sample. And that worked really well, again, because of that taxonomic difference. The blocking primer was able to only prevent sea lamprey DNA from amplifying really specifically and really well, and still allow for all the other fish DNA to amplify. So that was very successful. And once we implemented that, it was pretty easy to only get the host fish DNA. So the DNA, I say host fish is the prey that the fish that the sea lamprey were feeding on. So the prey of the fish of the sea lamprey, we also call it the host fish since it's considered a parasite as well. Uh,
Mhm.
but it was quite successful in determining, or at least getting a large chunk of our DNA to be DNA from whatever fish the sea lamprey were feeding on. So that was pretty cool. It was not a guarantee that it would work super well, but it was nice to finally get the results back, and everything looks fairly clean. It's sort of a messy process with DNA metabarcoding. A lot of things with DNA and genetics are just messy in general. You're just working with such it almost feels like magic in a way, right? You can't see the DNA. There's nothing in front of you working on. You're just putting reagents into tubes and mixing them and put them in a centrifuge, and then you're adding these other things and you put it in this big machine and it does some magic, and then you have your DNA, and then you do all this other stuff. So uh, all of the processes along the way, though, it can be really sensitive, and there's a lot of stochasticity in the process here, so it can be a little messy. So coming out with the results from this looking fairly clean was pretty satisfying.
Very cool. And
And yeah,
are you guys able to identify multiple species within, say, an individual sea lamprey, like multiple host species in the DNA samples?
yeah, that was one of the questions that we looked at, which yes, we were able to, which was very exciting as well, because there was there was sort of there's the two life stages that we were interested in looking at, and we are interested in different questions with them for different reasons. So we have the parasitic phase, when the sea lamprey go into the Great Lakes and they're feeding on everything. They're acting as parasites, and this active feeding phase. This is when they can get captured by commercial fishing vessels. So this is where
Mhm.
we can get sort of those direct observations of what they're feeding on. So the question in this phase was your question: can we get multiple species detected from a single lamprey? Because if we capture a lamprey, we already know what it's feeding on, right? If we catch a sea lamprey attached to a lake trout, I don't need to do a genetic test for what it's feeding on.
Right.
But if we can detect multiple species, or as we put it, sort of a feeding history in a way, then those sea lamprey that are captured on a lake trout or some other fish are still useful in a genetic testing context. We can
Hmm.
still perhaps look at what it was feeding on before the lake trout. How far back can we look? And all of that is still useful. So that was the one phase we're interested in, and then the other phase is when they are considered adults. Terminology is kind of confusing, but they're considered adults when they migrate back upstream to spawn. So
Right.
this is which I forgot to mention, sort of this phase in the life cycle. But when they do migrate up to spawn, they are no longer feeding. So they sort of undergo this physiological process where their stomach, I guess it's not technically a stomach, their intestine atrophies and sort of goes away or shrivels down a bit, and they cease feeding. So this was a really interesting area for us to look into because capturing sea lamprey in this context in a river takes away sort of any potential sample bias that might occur with using a commercial fishing vessel, right? Because these fishing vessels are going out looking for lake trout, looking for whitefish, looking for something else, and there's a certain, uh, I don't want to be too harsh, but potential bias that can come along with sort of this targeted mission where the only sea lamprey you catch are based upon this directed initiative to try to catch a specific species. Whereas when they're all just migrating upstream, just a whole bunch of them, if you capture them in a net or a barrier migrating upstream, it's sort of this more comprehensive, less biased sample size, more representative of the population as a whole, right? Because it's sort of random lamprey that you could capture in your net there. So the question here was, say, sea lamprey had been migrating upstream for several days. We tested a range of up to 30 days. If they had not fed on anything for 30 days, are we still able to detect DNA in the stomach at that point? Because then those adult lamprey that are migrating would be useful for genetic studies. We could capture that, and that'd be really important. And it turns out they were. There was a bit of a decline as the days increased a little bit, but not as much as we even expected. There was still this was tested in the laboratory, so we we specifically let sea lamprey feed on a fish. We let it not feed for 30 days, and then we looked to see if we saw the DNA of the fish we know that it fed on. Um, and yeah, even a couple samples up to 45 days still had detectable DNA in the stomach that they had been of the fish that they had been feeding on, which was pretty cool as well. Then we tested it with a couple different temperatures as well to see if there was any effect on perhaps higher temperatures increasing the metabolism of the sea lamprey, therefore increasing the digestive rates, therefore degrading the DNA even more, and perhaps that would make it more difficult to detect. We saw a little bit of that as well, but still, even at 30 days and at 15 degrees Celsius, which was the highest temperature we tested, water temperature, uh, we still saw some detections of host fish in there. So that was really cool, and it sort of expands the applicability of this method in the future if we want to keep using it on different river systems and tributaries in the Great Lakes in the future. That just allows for more range. We can test it sort of probably throughout the season at different temperatures, etc.
That's so interesting. I feel like I can almost already see the way this project has evolved, because I'm sure that it started one way and it's gone so many different ways the more you found out about all of the different kind of facets of it. I have two more questions on the molecular side, and then we'll kind of transition into maybe some other things or wherever we want to go. But one is, what is the most unique thing you found in a DNA sample of a sea lamprey? And then two is, and this is kind of a yes or no, but is it possible, say a lake trout ate a whitefish, is it possible that you're getting DNA from a whitefish in that blood of the lake trout? Maybe not because it's eating it. I don't know. Just curious.
Yeah, that's a yeah, tackling that second one first. That's an interesting thought. There is, I would say, probably not likely, although I did a little bit of digging into, because think about how that might work is you have DNA of your whitefish in the stomach of a lake trout. Is there a way that the DNA of that whitefish gets into the blood of the lake trout?
Yeah, I know.
And there is actually
Hmm.
certain processes by which DNA can go into the bloodstream of whatever organism ate it, and there's been some studies on this with humans as well, because there's been interest in if humans eat something, does the DNA of that thing get into our blood? So there's actually sort of research more in that realm, because humans are going to get the research first on
Yeah,
a
yeah,
lot of
yeah.
topics.
Hmm.
So there has been like some NIH studies on that, and it does happen actually. So
Hmm.
and it's such a sensitive process that I would say it's not completely out of the question that something like that could happen.
So
So
that's the next
yeah,
step for your guys' project is
yeah, that's the next investigation. So
there
thank you,
you go.
Jake. You just gave me a bunch more work to do, but
Back to school, man.
Make
Back to
sure to
I
school.
got a second degree looking at that.
PhD,
make
PhD.
sure to cite
Exactly.
Jake and study.
Yeah, Jake.
Sample me.
Project design, Jake
Yeah.
Yeah.
Johnson.
Uh,
So no, very cool though. It's a
mhm.
very good idea. And yeah, the most interesting thing that comes to mind actually wasn't specifically a DNA. We did something that was cool DNA wise, and then I have a non-DNA interesting thing
Mhm.
was on the DNA. It was interesting that in some of the adult samples, so this was this was outside the lab, so that we had our laboratory test. That was sort of my second chapter, and the third chapter was applying all this in the wild. So we looked at about 1,000 sea lamprey samples in the wild, and some of the adult migrating sea lamprey that hadn't fed. Some of those samples returned four, possibly even a little more species in a single sample.
Mm.
So uh, that was quite interesting for something that we didn't even know up front if we'd get any DNA in those non-feeding sea lamprey samples to have them returning. Even one was interesting, but two, three, sometimes four different species was pretty pretty interesting as well. So that was very cool. It's it's sometimes we're looking at it's contamination is always a potential issue. When I'm in
Mhm.
the lab, it's super sanitary. You have to constantly be wiping things down, using a fume hood, etc., gloves. So there's contamination that can happen within just the genetic process as well.
Mhm.
The way that all that magic happens under the hood in those big machines. There are ways that the readings can be slightly different because it's just so sensitive. So it's possible contamination it could account for some of those, but I saw I saw enough samples with multiple species in them that it was unlikely contamination for all of them. So I thought that was pretty interesting to see.
Yeah, that is
Uh,
interesting.
and then just another side note that was interesting too, not on the DNA side, but one of the digestive samples, I didn't do the extractions. I did the extractions. What's the word? When you dissections. That's the word. I
Mm,
didn't do
there
the
you go.
dissections myself. That was up at Hammond Bay, but when one of the technicians opened up a sea lamprey, she saw a fish egg in it. I think it was a whitefish egg, which was
Oh,
super weird. We don't
yeah.
we don't know how that really got in there. It could have been perhaps a whitefish that had eggs in her, and then the feeding of the sea lamprey ingested an egg some way like that, as opposed to them feeding along the bottom or something. But that was pretty interesting as well. Did not expect to see any eggs. So
Yeah, it's definitely That would be like, what? What's going on here? I mean, I almost feel like it's
Yeah.
like a sea lamprey coming up to a female. They're like, oh no, here's my. I don't know, but um, Dean, you got anything here? Because I got another question if you don't.
No, go ahead, Jake. You're right.
So, I know that's the more molecular side of things, Jurassic Park-ish, if you will. So let's go over to the. I know we had talked about you had done some conversations with fishermen. You had gone out and talked to fishermen. What was their reactions when you asked them about sea lamprey? And was it common that these fishermen on the sea, what were you at Lake Michigan or wherever you were like, what was kind of their reaction to your study and what they've seen so far?
Yeah, so that was a little before I did my project specifically,
Mm.
where I worked with it was under the University of Illinois, and I did creel surveys where I went out to fishermen on Lake Michigan, and I essentially the goal of that was to gauge what's called fishing effort. So the amount of time, the amount of attention, the amount of money that people spend fishing over the lake, because fishing effort is an important factor to estimating fish populations and other variables, right? If populations go down by a little bit, but fishing effort went up by a lot, that could tell you a different story than if populations went down a lot, but fishing effort didn't change, right? That's two different sort of stories there you have to figure out. So talking with fishermen was an interesting experience. They're an interesting bunch. You get a whole variety of personalities there.
Yeah.
Some of them are extremely interested, and I could talk to them for a long time. They'll see me coming up to them. They'll be, "Oh, you work with so and so. You're the fisheries guy," and they're really interested in the lake, and they're interested in all these invasive species. There's more invasive species than just lamprey, sea lamprey in the Great Lakes, but they're interested in all the stories behind all of this and interested in protecting and what we do. And then there's others that want nothing to do with me. They probably think I'm the DNR and I'm going to check their fishing license or something, which I'm not. But
Yeah.
it's interesting always to talk to them a little bit too. But I think I think an interesting takeaway from talking to fishermen is that all the fishermen have sort of their own story on either what's wrong with the lake or why they're not catching any fish. There's always something like, oh, back 10 years ago, I'd be catching fish all the time off this dock here. I blame all the alewife or something, or I blame the sea lamprey, or I blame the DNR. They're not stocking enough. So everybody has their own story kind of on what's going on. And yeah,
Hmm.
they're all right, I would say, in one way or another. I'm not here to say that this guy's right, this guy's wrong. The fishermen are out on the water more than any of the biologists are. Like we have our field season, but those guys are out there every single day, every single area. They cover more ground than we could, and they've got their eyes on the water more than we do. So it's always interesting to hear the stories that they have and what their experiences are.
Yeah, we had my dad on an episode. We were talking about him just as a fisherman, and one of Dean's points in talking to him was that, these fishermen and hunters, people who are out there out in the out in nature all the time, like you said, every day. They're the ones who are seeing it. They're the ones who are getting this, up close and personal perspective. And they may not be doing the molecular studies that you are, but they do have a very interesting perspective, and it's definitely worth getting their opinions and feedback. So that's cool. I like that. I didn't realize that was not part of the same project. Again, we've been friends for a long time now, so it all kind of mixes up together. But
Does
um, very cool. Any finishing questions, Dean? Any finishing questions for Connor on this subject?
I just wanted to ask you a general question about your
Yeah.
research, just
Yeah.
out of
Mhm.
just out of curiosity, and that was um, what have you enjoyed most about your research? And this could be applied to research in general. It could be specific to your project. What have you enjoyed most, and what do you possibly dread about the research? I'm
Yeah,
curious.
I would say something that I've found really cool with doing, and this could really apply to just research in general, probably, but also to what I've done specifically is you get so enveloped in just this very niche specific topic. And because it's new, it's research. We're answering questions that didn't previously have answers, or at least trying to get better insights into questions that didn't have answers before. That when you're at the forefront and of a project, doing your own research, master's, PhD, or afterwards, it's really cool that you just become sort of the world expert in a way on a super 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 niche tiny thing. Even certain questions and topics that I would might understand this very specific thing a little better than people who have been in the field generally for a long time, right? Anything related to sea lamprey specific blocking primers, I would probably be the guy for that. It's like
Right.
not like anyone's gonna really ask me about that in my life ever, but it's really cool to be like, hey, I'm sort of expanding the general knowledge that we have
We're
about
asking you
this.
right now.
Yeah, right. There you go. That's true.
You just
That's true.
talked
So
about it
there
for
you go.
50
I
minutes.
know.
Yeah.
Very true. Yeah. So that's it. So I think just having a super hyper knowledge about just a super specific topic and having real world impacts on that. It's
Definitely.
any research. The goal is well, not any research, but a lot of research. The goal is to sort of have some real world impacts here or there, or change the way that somebody thinks about something, and that's having that impact is really cool. So I think that is probably certainly one of my favorite aspects of it, is that you had this direct impact on something. So if I can improve the way that sea lamprey are managed or controlled in the Great Lakes just a little bit more, I think that's really, really cool. Of course, there's a lot of work that goes into that too. So probably my least personal favorite side of things was just I really got tired of a lot of the wet lab stuff. I didn't mind the writing, I didn't mind the data analysis. I actually really liked both of those things, but I think being in the lab, just sort of with pipettes for 10 hours, go
Mm.
to bed, do the next thing the next day, you know, uh, that got pretty tiring after
Yeah.
after not too long. But I still had a lot to do, so it was pretty tiring after a while, and it honestly it sort of shaped the direction of where I want to go now. I'd much rather do the computational data analysis side of things as opposed to being in the lab. And nothing against anyone who does that or enjoys that, it just wasn't for me. So yeah, probably my least enjoyable part of it.
Yeah, and that segues
Sure.
perfectly into my final question about this. What's next for you? So you obviously finished your master's program. Was it beginning of this year, end of last year? End of last
Yeah,
year, right? Yeah.
yeah.
Um, what's next? I know you like data analysis. Is that where you're looking to go? Do something in that realm?
Yeah. I really liked the computational side of things. So, specific to genetics, it's a field called bioinformatics, where the pace at which genetic technology has improved over the past couple decades is astronomical in a way. It's outpaced even. There's sort of this, I believe it's Moore's law or something, that sort of has this exponential growth of technology, and genetics has improved far faster than even that. Just with, if you compare, in 2003 I believe 2003, we finished the sequence of a human genome for the first time. It was a huge project, took several years, and we sequenced the human genome. Cost 3 billion, I want to say,
Mm.
uh,
Well,
and 3 million, something like that.
A
that's a
lot.
little difference, but
I know.
A
I
lot,
know. I'm,
man.
I'm, I'm fumbling the big numbers now, but it was. I want to say 3 billion, and currently it costs. You could sequence a human genome for a few $100.
wow.
And you can do it in a day. And the pace at which its technology is improved is incredibly fast. And what comes with that is a whole ton of data. There is a lot of data that goes into the analyzing of all of the genetic sequences that are output. And that was a part of my project that I really enjoyed. Sort of, it's a lot of coding, sort of putting together pipelines that can take the raw data from the sequencer and they filter it one way and they check for the quality and then they remove some things and then they check for quality again and they filter this, etc. And I just ended up really liking that sort of realm and that area of research. So looking at something in that direction. So looking at bioinformatics now as a career option for me. So likely won't be with Sea Lamprey. There's not a lot of jobs out there
The
specific
mystery
to
ends.
Sea Lamprey. Yeah, uh, probably not even fish in general, but there's a lot of this work, of course, with humans. There's things with personalized medicine, personalized genomics. I think that all of that is really interesting as well. Also with microbiome work, so gut biome work is pretty interesting as well, and that specific genetic work is similar to the DNA metabarcoding that I did myself and mentioned. So the cool thing about working with genetics with one species is that it more or less fully transfers over to another species, right? It's just all A, T, G, and C in one species, and it's the same in the other. It's the same code, it's the same language. So it can transfer well into other sort of disciplines or other species at least. So working on humans versus fish will be the same sort of computational knowledge. So yeah, looking at jobs at that, it might lead me to industry. There's certainly a lot of interesting industry positions. Might lead me to an academic position. There are labs that will have PhD students and master students in the lab, but they'll also hire staff bioinformaticians, in this case, staff researchers, to just work on the computational side of everybody else's project, and sort of as needed, and help with ideation and sort of connect some dots between what needs to happen on the data analysis side versus whether that's applicable given the sort of research idea that they have, etc. So something along those lines. But yeah, I think I'm moving into the sort of computational data analysis realm of genetics.
Awesome. Well, Connor, as we approach the 1 hour mark, we want to thank you for being on the podcast. Very interesting insight into Sea Lamprey and everything with your project at Michigan State. I learned a lot, and I've already known a lot about this project, so it was really cool talking to you. Would you mind doing another trick on the way out? You're not a pony, but can you do the one where you like shuffle the cards up? You know that one.
The spring,
The
the
spring. Yeah, he's got
classic.
words for it.
Of course,
Jake's
A
the
magician
word,
in the
the
lab
word
and
for
with
everything.
the cards.
All right. I know. I had plenty of dexterity practice for my pipetting that I did
Yeah.
there. So, see here.
Impressive.
Whoa.
I got to do the sound effects too.
Beautiful.
Yeah.
Beautiful.
Do it.
Everyone, make sure you check out Square Up. Connor, where can people find Square Up if they want to check it out?
Plug
Square
away.
up, spelled square up. It
At
is
Square Up on Instagram.
at Square Up Cards on Instagram. We got a TikTok too, unbanned now, so we're back. Square Up Cards as well. That focuses on tutorials for beginners. So if you see me doing this stuff and you think you could never do that, you can, and we can teach you. So we teach a lot of the stuff. But yeah, Square Up Cards would be where to find us, and then on YouTube as well.
Cool. Awesome.
Cool.
Well, appreciate you going on the podcast. I think today we learned a lot about how conservation goes into so many different areas, niche, broad, whatever it may be. But there's a lot of people working out there doing a lot of different things that all impact conservation as a whole. So, Connor O'Kane, shout out to you. Thank you for joining the podcast. Dean, great, another great episode. Having another guest on the podcast. That was awesome. For everyone listening, make sure you give us a like and subscribe to the channel. We appreciate you all, and we will hear you next time on the Chasing Wild podcast. Thanks.
Thank you.
Thanks, guys. It's fun.